And we are on. So welcome to everybody to this new instance, this new repetition of the International Legume Society webinars. I'm very happy to have here for the very last webinar of from this year a colleague but also friend of ours, Carlota Vaz Patto, from the Universidade Nova de Lisboa. Today we are going to talk about Lathyrus or Cicerchia, as is known here in Italy. And please, Carlota, start, and then, as usual, we'll have a chat after the webinar.
Okay, thank you, Nelson. I will start sharing my screen. Let's see if I do it well. Okay, can you please confirm that you are seeing it? Okay, good. Just organizing a bit my screen. Okay, so today I will tell you a little bit about the work we are doing in my group, the work related to Lathyrus, mainly Lathyrus sativus, graspy. And I will show you how it has been a little bit tendrillous path, very connected to the tendrils of this plant. So let me see if this moves. Doesn't move. How can I do it here? Yeah. So I will start this talk by imagining a world where all the plant breeders would have all the tools that they will need to produce beautiful varieties. And so, of course, they will need all the genetic material available and diversity at their hands that would allow them to produce the best varieties in very easy ways. And that it will be also a world where the farmers will be happy and convinced and to use this crop or these new varieties in such a way that they could bring their materials to the markets, and they will be easily sold because they would represent the interests also of consumers and all the stakeholders in the market. Unfortunately, and as you know, because you work or have an interest at least on legume species, this is not the case in many legume species, especially not on many underused crops. And this is what we are going to tell you about that is happening with this particular legume crop that I work with, graspy, Lathyrus sativus. This is a very nice species, very resilient, and that has a lot of benefits. However, so it can stand a lot of you can say adverse environments, different stresses, but and because of all of that is considered a model crop for sustainable agriculture. Nevertheless, this plant in particular, on the places where it is produced, especially in the Mediterranean region, has been facing a lot of constraints that, especially in this region, are related to the increase of the temperature, weather, environmental temperature, that is going faster in this Mediterranean region compared to the rest of the world. And also with a lot of events of not only drought, but also heavy rains more recently. And this is complicating a lot, even the development of this highly resilient plant or species. So the problems are mainly this, so that there is an increase on drought events and unexpected floods. And this leads then also to an increase, because especially the increase of temperature, on increase on the frequency and intensity of pests and diseases outbreaks. And our farmers are already suffering from all of this. The farmers that we know and we work with on graspy production. So there is this fundamental need that even on this particular hardish crop, we need more productive, resilient, or adaptable plant varieties that can cope with this climate change negative effects. Okay, let me see the point. So this is a tendril. It's not a clear, a real tendril. It was construction I did, but I will use it as the table of contents of my presentation. Okay, and I will try to explain you why the path of our work on Lathyrus is following some way similar to this tendril. So just as a little animation, you can see this is the name of my group, PlantX, and we have been curling around on the research with Graspy. We started maybe around 2005 already, and at that time with a great collaboration with a group of Diego Rubiales in Cordoba in Spain from ILSW, we started to analyze an Iberian collection of Graspy, Lathyrus sativus, and also Lathyrus cicera. And at that time, we chose to do mainly work with the fungal diseases we were more used to, and in my case was rust and powdery mildew. And we did a very detailed characterization of the resistant mechanisms that were present in these two species to these two different pathogens, identifying very interesting levels of partial resistance that later on also changed the way we were seeing plant resistance to different other pathogens. A little bit further on a couple of years later on, we decided that we should increase the diversity of material that we were analyzing. And so we started to collect and gather a worldwide representative of the diversity of this species, Lathyrus sativus, coming from different parts of the world. And for that particular aspect, no, we didn't travel all over, would be very nice. But we used instead the connections to the different germplasm banks in different parts of the world. And we gathered this quite nice and representative collection of the diversity existing in this species. I've mentioned here the year 2012 in more detail, because this was the time that we also did, in that case, we travel around Portugal, and I will tell you a little bit about that later on. But we start connecting with the different farmers that were still producing Graspy in Portugal at that time. And I didn't have that connection until then. So it was really nice to see and to understand that there were still a lot of people working on this species, cultivating this species, keeping their own traditional land races. And we started also to understand what were the problems of these farmers in particular. At that time, we were very interested to develop genetic resources for this species because there were not many. And so in those years, we focus a lot on developing molecular markers, on studying the diversity of this collection and things like that. But later on, and so we follow this path, a few years later, we started really work more close to the producers, the farmers, the processors. And we did that on the most important region for Graspy cultivation in Portugal, that is Alveias region. And we counted with the collaboration of the Alveias municipality and some farmers of that region. And indeed, this was a time-changing, a life-changing event, because we start focusing on the traits that were really important for the farmers and the problems that they were facing at that time. So these were the first steps for us on participatory research with Graspy. And so we started by characterizing the collection we gathered from all over the world for resistance to Fusarium wilt. That was one of the first concerns that the farmers brought to us. Then later on, we moved to adaptation we wanted to see if we could find already from the material we gathered from all over the world, if we could find some accessions that were already answered also to the increased temperatures or so the changing of the environment under Mediterranean conditions, so adaptation to Mediterranean conditions. And this followed also more detailed analysis on quality traits related to end users. We came back again to powdery mildew and rust because these were the first pathosystems we studied. And now that we had more diversity, we also want to see if we could find some nice materials, sources of resistance to them. And then finally, we studied drought and flood in quite a lot of detail. And we are presently also focusing on aphid resistance. In the most recent years, we can say that we have now a GRASP living lab established, connecting the different researchers and stakeholders involved on this research. And we are counting now with a DECA, that is a local development association. We are counting with the collaboration of many farmers, small processors, and we are also involved on a citizen science initiative more close by to Lisbon, so to our location in Oeiras. And in that, we count with another municipality, and this is the support of the Oeiras municipality. Okay, so taking this into account, I just want to present you the GRASP Living Lab as it is right now. It is focused mainly on Alveias region, as I told you. They are called themselves the capital of GRASP in Portugal. It is a very nice area. And these are us, okay? This is the group of people we are working all together. You have here different researchers, different farmers, different small processors, the municipalities, and the training schools. And we are all working together to develop a more resilient resistance to research in grasping. Presently, we are funded by a European project, Live in Food, but also at the citizen science level, we are funded by another European project, Impetus, besides the municipality support. Okay, so what do we do with this very diverse ponder, as we normally call it? These are seeds of different graspy lines. And what we decided was to exploit this natural variation to understand the genetics behind all of the traits that are important for our farmers. We are doing that by using this worldwide collection that I told you already, that we gather. And our first approach was like using a genome-wide association study. Just very quickly, I can tell you that our collection has this constitution, it's constituted by different lines from different parts of the world, mainly Mediterranean, because that was a little bit biased since we were here. But we also have South Asian, Eastern Asian, Sub-Saharan Africa, North and Central Asian, and then a few from America, and a few that we don't know exactly where they are coming from, but they are quite interested to keep on our analysis. And of course, we have representatives of the two main ecotypes that exist on graspy, that are the Mediterranean ecotype, known by these big seeds, light colors, and white flowers, and the Asian ecotype that is characterized by much smaller seeds, dark colors, and blue flowers. These are the two main ones. Of course, there are other ecotypes in between. So what do we do when we are talking about genome-wide association studies? I will go very fast because but I would like to explain it a little bit, because then I don't need to repeat it every time. I will tell you about the work we are doing in different traits. We are interested on traits that are normally, all the ones I mentioned to you, are normally controlled by many genes. And so they represent a continuous variation of phenotypes. And because they are also, and this continuous variation is also due because they are influenced by the environment. So what normally we do, can you see my pointer? Yeah? You can see the pointer or not? Yes? Okay. So here it's a representation of our association panel, our collection of sessions that is, in this case, at the time we did this, about 200 graspy sessions from all over the world. And what we did was that we scored them at genotypic level using different SNP data. And we scored them also for the different traits at phenotypic level. And then we are just trying to look if there is a significant difference between the mean of the phenotypic values between the different genotypic classes. And we do that by statistics, and this is just an example of how we can do that. So if we, in SNP2, we have a particular base in the two different lines, a C and a T, and the mean of the lines that have a C is about 18, and the lines that have a T is about 110. It means that there is a correlation between the presence of the T and the increase of the mean. So we use from all the we analyzed this panel with different approaches. We used genotyping by sequencing and DartSeq, and we gather about 26,000 markers, but from them only about 5,600 are of high quality, and so they were kept for this association analysis. And then, of course, the traits we are interested in can be very diverse, from stress resistance to adaptation and quality. So then we perform an integration of these two types of data to detect if there is any SNP that is highly associated with the variation of your trait, and then see where is this SNP into the genome. And we do that using univariate analysis, and then we localize and try to find out a biological meaning for candidate genes in this particular SNPs within candidate genes. I have to say something here. Okay, at the time we did this, there was no how do you say, reference genome for Lathyrus. So we were using a close by species, that is P, and that was the best annotated we had at the time, genome we had at the time to use in this analysis. What makes it a little bit challenge, but we managed. So I will tell you how we did it with a few examples. So I said these were the traits we are interested in, but we will keep on just with ones you see over here. I will give you a little flavor of all of this, but not too much detail not to get you too tired on it. So I will start with Fusarium wilt. This was the first trait we were using on this type of genetic studies. And this is the work of one of our previous PhD students. Now she's a postdoc in Switzerland, Anna Margarida Sampaio. And what she did was she wanted to characterize this collection we gathered for resistance to Fusarium wilt, and to identify the mechanisms that were behind this, if there were different mechanisms behind this resistance. So she used a collection of about 179 accessions of this grass pea, working with 1 week old plants. She used the root dip inoculation. And then, of course, with different several repetitions and several independent inoculations. And she measured this for different traits associated with the disease progression or intense Fusarium wilt phenotyping. So that is disease intensity, disease progression rate, the maximum disease intensity at 30 days, and then the area under the disease progression curve. Sorry for the typos. This means that this phenotyping took at least 30 days to complete in each inoculation step. Okay, so what did she observe when we are looking at the diversity of resistance levels? She saw a very significant effect of the genotype, so there was clearly genetic differences between the different accessions. She also saw a quite wide range of responses, but indeed, and if you look at this particular 3D figures, they are exactly the same, but they are colored code related to if the seeds of that accessions were dark or light in this first over here. And in the second one, if the seeds of that accession were large or small. And what we see over here is that the highest values, especially for our DPC, but also for DY1, DY30, are for the dark seeds, the dark seed types, and the small sized seeds. Okay, so indeed what she understood was that, or concluded was that the lighter and larger seeds were more resistant. And then that are the ones that you can see over here. So the orange that never goes until there, or the red that never goes until there. And there were a broader response, different types from highly susceptible to resistance within the smaller and darker seed types. Okay, then When we correlate the disease levels or resistance with the origin of the sections that we had in the collection, because we knew a little bit about where they were coming from, it was very interesting to identify that these ones over here that were the most susceptible ones were from Ethiopian origin. So this is something to keep in mind, okay? It really looks very interesting, the material that comes from Ethiopia. In this case, they are highly susceptible, but in other cases, it's not what is happening. So just bear with me, and I will give you a little bit of other examples that will explain you this. So after this, Margarida was interested to know what was the genetic basis of this particular resistance or mechanisms that she detected, or the resistance levels that she detected in this collection. So she started to do, or she did the first genome-wide association study in Graspi, and this was related to Fusarium wilt resistance. The first thing she had to do was to study the genetic structure of this collection, and this was the first time that we also did it in this detail. And what we observed was that when we look at the population structure, and in this case, this was the most logical K, it was 2, so 2 main gene pools existed on this population. And what you can see over here is the seed colors and sizes. And you see that gene pool A, so the red ones, the sections that are light and large, or that have light and large seeds, are normally only from the first gene pool. And then when we are coming to the darker and smaller seed sections, it's much bigger mixture with the second gene pool. When we look at the principal component analysis based on these same molecular markers, indeed we see that there is always much less, or the clustering is much bigger when we are looking at the light color seeds. So here it's a representation between light and dark colors, and the lighter are much more clustered than the darker colors that are a little bit more dispersed. And in this case is related to the, sorry, that is covered, but it has to do with the size. So in this particular case, the bigger sizes are also more concentrated here than the smaller sizes that are more dispersed. And when we look at the geographic origin of these materials, we also can see that the red colors over here are mainly Mediterranean material, or European. Yeah, you call it European. It's a little bit different classification in here, but they are much more clustered than the rest of the other origins, like for instance, Asian material. Okay, so moving forward, she then got this data on these 3 different traits that she measured, so ODPC, DY30, and DIR, and she performed a GWAS study based on the number of SNPs that I already identified at the beginning. And she identified 17 different genomic regions associated with one of these traits, and that were located mainly on chromosome 1, 6, and 7. And then, of course, she also looked for candidate genes that were in linkage disequilibrium with this particular SNPs, and they were involved in secondary or amino acid metabolism, RNA, and transport and development. This is not the most important thing, because here what we wanted to see was if indeed there was an oligogenic control of this trait. That was the case. There was also a quite big correlation between the different traits. So also it was not unusual that the SNPs would be common in many of the cases, the associated SNPs. And this gave us already a few, the first steps, you can say, for the development of a few molecular markers that can be used in the future to select for resistance to Fusarium. But not only this, also the characterization of the collection in this detail gave us also identified also important sources of resistance to Fusarium that can be used on breeding programs. Okay, so when we look at powdery mildew, and this is a little bit different story, but much more simple, you could say. In this particular case, we were also working, and this is the work of another previous PhD student, now a postdoc in the north of Portugal, David Martins. And he was interested to study the resistant mechanisms of grass pea to powdery mildew. The first pathogen he studied was RACP-PC. That was, it is the causative agent of powdery mildew in pea. But he also looked at RACP3 that is the clover, causing a pathogen, causing powdery mildew in clover, sorry. And why? This is quite important because if we have a plant that is affected by RACP-PC, that is the P pathogen, and P is now also, there is a report that it can also be affected by RACP3, and that is RACP3 is overcoming the main resistance gene ER1 of P for powdery mildew. We can start thinking that maybe this pathogen can start being also a threat on grass pea. So that's why we studied both at the same time. And what we identified was that indeed, RACP3 caused much more disease severity in grass pea than RACP-PC. And unfortunately, or not unfortunately, this is the fact, there was much more partial resistance to RACP-PC than to RACP3 in this collection. Okay, so all the sessions we studied had a competitive interaction between grass pea and the pathogen. That's why the infection types were 3 to 4, with significant disease severity differences among the genotypes. Some of the cases I said had partial resistance, so this means that they have a reduced disease severity but a high IT or infection type. But there was not a clear, strong correlation between the two phenotypes. It was about 0.58. Nevertheless, there were multiple resistant accessions. So multiple means that they were resistant to both pathogens, and they were Ethiopian origin. So you see, before they were not the best ones, but now they are really very interesting material. And so what happens later on, or we can see is also here this part about the Ethiopian material. We don't see it very well because the colors are not so good, but the Ethiopian material is red. So, sorry. And sorry for the people that don't distinguish the green dots, but this was the picture I had. The green dots are mainly here. They represent Ethiopian material, and that means they had the lowest disease severity scores for RACP-PC and RACP3. So that's quite an interesting source of resistance for breeding programs of grass pea. When David started analyzing or performing GWAS studies in this material, he saw that, of course, because the correlation was also not big between these two phenotyping data, only one of the many SNPs that he identified associated to this disease resistance was shared between the two of them. But this was not surprised because of no correlation. So, and these resistant candidate genes were, many of them were resistant proteins, and so they were biological relevant candidates that we could consider for future marker resistant selection or other types of selection. So when we look at the drought and flood, and in this case is the work of a PhD student, Matilde Sanches, she was interested to characterize this collection for drought and flood resistance. And why? Because we know that this material is quite, or this species is quite resilient, and it is always said that it is resilient, but then there is not a lot of data on this type of more detailed analysis. And that's why Mathilde started working with that. She had this very nice approach to study 3 different conditions: well water, water deficit, and water logging conditions. So all the collection was analyzed into these 3 different conditions. She used different physiological traits, and she measured about 20 different traits based on gas exchange, chlorophyll fluorescence, relative water content, biomass, partition and production, and pigments content. So you use different equipment to do all these measurements. This was a very long phenotyping procedure, but she managed and she got a lot of data at the end. Here you just represent principal component analysis, where you can see what is these three colors clusters represent the conditions, the sessions in water deficit conditions, in well water conditions, and in waterlogging conditions. You can see that the water treatments, you can say, cluster a little bit apart, but with quite a big overlap. The first four components explain about 80% of the total variability. You can see that the first separation is based on the relative water content and on the partition of biomass root to shoot. And then also on the gas exchange that you have also over here. And then the second separation, that is more what you see within each treatment, within the collection, are more related to pigment contents and chlorophyll A fluorescence. And then the last component is more related to water use efficiency. There was a high correlation among the groups of traits that were expected. And some of the traits, like total dry biomass, is more explained by the genetic component than by the environmental component. The main conclusions was that the light and large seed, mostly of Mediterranean and East European origins, produce plants with high total dry biomass, but this is also happening in other species like pea, when the large seeds are normally producing bigger plants. But then also interesting to realize that mainly the dark and small seed accessions showed advantaged physiological response to both water deficit and water logging. And this was measured through pigment contents, root to shoot ratio, and water use efficiency. So what she did after this was to identify the most contrasting accessions, a group of most contrasting accessions, quite complicated because she had a lot of traits to measure and integrate at the same time, but mainly based on the difference between the values on stress conditions and under control conditions, well water conditions, and based on, so these delta values, based on water use efficiency for selecting for water deficit tolerant and water deficit susceptible, and using photosynthetic rate to select for water logging tolerant and water logging susceptible. She identified even accessions that were a double tolerance, like this accession highlighted here in green, that is tolerant to water deficit but also tolerant to water logging, and the other way around, a double susceptible accession over here in red. And then she also realized that the main water deficit susceptible plants were the big ones, so the big seeds with light colors, and the tolerant accessions were normally in both treatments, were normally the smaller seed and darker colors. So this is also interesting to keep in mind. Okay, so, and this, just to finish this part, what we say is that incredible times are ahead, because until now we used a reference genome from pea to be able to do our the results I show you for the genome-wide association studies on grass pea. But very recently, two other genomes have come along now on grass pea, and more recently a new reference genome with a chromosome scale, where we also collaborated with, is now published, and it is really an incredible tool for us, because I don't know if you realize, but some of the markers we had on our collection were not mapped, so we had like a unmapped chromosome at the end, and now we are able to map much more of our screened SNPs, and also to have a bigger number of molecular markers from our genotyping by sequencing approaches. Okay, just to keep in mind that Ethiopian material was the most resistant to Erysifi PC and Erysifi trifoli, however, was the most susceptible for Fusarium oxysporum, but nevertheless is the less affected to contrasting water stresses. So really just to highlight that how important this can be if we know where the material is coming from and where to look for interesting sources for our breeding program. So unfortunately, in this for Mediterranean breeders, that farmers are more interested in white colors and bigger sizes, the darker colors and smaller sizes from Ethiopian material could be maybe a problem, but nevertheless quite interesting materials to start crossing from. And so that is what we are doing now. I told you that we did this tour around Portugal to get these traditional land races from different farmers. We did that tour not only in the Alveias region, but also in the mountains near Lisbon and the mountains in Algarve, that are the three main regions of production of grass pea. And from that, we gathered this collection that we are working now with collaboration with different farmers, where for several years we had multiplied this material on the field, and we gathered the evaluation done by the farmers on this material. So what were their opinions on these materials? And so we start cross-breeding activities now, not only based on the participatory selection, but that is also counting for the material we are crossing now, but also all the other traits that we measured under more controlled conditions. And hopefully we are now multiplying the first generation of segregating populations. And we know in the future we will contribute to the development of new varieties, more productive, more stable, and more interesting for our farmers. And that's it. Thank you so much for your attention. And these are the funding bodies of all of this work. Thank you. I will be happy to take any questions now.
Thank you, Carlota. Your presentation was on point, pretty interesting. So before having other questions from the group, I have already an inquiry from the chat. Somebody was asking if you are okay with distributing the presentation.
Yes, no problem. No problem. Yeah.
Okay, you can send it to me and I will upload it on our website.
But nevertheless, it is taped, so it will be there also, right?
Sure. Then I will also share a link for everybody, as since a few months we started to share with everybody. We have a feedback form that you may want to fill to give us some feedback on how we are doing. And if you're okay, if you click the link and the feedback maybe is in Italian, but there is on top right a menu for changing the language. Okay, so this raised hand, and then I see a question from the chat. Somebody is asking, do you plan to work on ODAP?
Yeah. Well, ODAP is a component that is quite important on grass pea. I have to say that until recently, I was thinking that not for Mediterranean and it's still not important on Mediterranean conditions, because under equilibrium diets, ODAP it's not a problem for our health. But if you are eating only grass pea for a row, in 3 months in a row, and this is your staple food, then you can become sick, of course. But there is still also something else, is the stigma. Even if this is not producing disease anymore, because people are eating a more equilibrium diet, for instance, in some Asian countries, there is still the stigma there. And so people are still afraid of eating grass pea if they don't know that they have a reduced ODAP content. So I agree that the efforts to work on ODAP, to reduce ODAP, are important. It's not our main focus, and it will not be, but nevertheless, we will measure it, of course, especially because it can be also correlated to other type of traits we are more interested in. And we want to clear out if this is the case. For instance, for drought resistance, there's a lot of literature that is saying that maybe this compound can have an effect or be a cause or an effect of this higher drought resilience. So we are, of course, taking a look at that, but we didn't do it still in a very systematic way. And
ODAP is
not with the same.
ODAP is the component linked to Lathyrus.
Yes, this is the component linked to Lathyrus, yes.
Okay. Okay, there's a question from Tom.
Okay. Hi, Carlota. Thank you for a very nice talk. I was surprised by one result that you described for Fusarium wilt, when you said the larger and lighter, especially the lighter color seeds were more resistant.
Yes.
That in our experience on peat, we find the opposite with, at least with Fusarium abenaceum. We find that the peas with the darker pigmented seed coats are more resistant. So I was surprised. Any thoughts on that?
It is. What I said was that indeed the larger and lighter colors are more resistant in general, and the darker colors are more broader responses, have a more broader response. Okay. What we think here is so we also have very resistant accession. We have resistant accessions with darker colors. We could think that maybe some components from this that provide these darker colors would have some antifungal activity, and that would be logical, I would say. And I think that's also what happens in different species. But in this case, I think maybe the most important part is the size of the root that is making a difference. So normally bigger seeds are also, we didn't test this. There's something I would like to test or to phenotype the roots into detail, that we could have this idea clear or not. But bigger seeds will also contribute to bigger plants, so bigger root systems, and somehow with more regenerative capacity. So even if it gets sick, it can compensate. But then the smaller roots, seeds or smaller root plants probably will avoid better this, but once they get the disease, they will die. So something like that. I think maybe in this case, maybe the root size will have a bigger effect than the components, chemical components that could make a difference. But I didn't test it. I think it's just an idea I would like to test.
Thank you.
Lathyrus.
Is somebody asking other questions?
I am Bruno from France. Just to say that if we want to develop this crop, I asked you a question about Lathyrism.
Lathyrus, yes.
And if you want to develop this, which could be very attractive for the agriculture with the global warming, etc., we need to secure the consumer. And that's why I was raising we have the same in fava bean. In France, there had been a strong development of low vicin, covicin varieties. So the fava bean consumption is increasing. So I think that with Jess, I don't know what do you say that in French is Jess. This will be mandatory to bring some reassurance to the consumer if we want to grow this production.
Well, I don't know, Bruno, because for instance, in Portugal and Spain, and even in Spain, there were big problems of Lathyrism during Civil War. The truth is that in Portugal, we never had it. And it is consumed, and in Spain was because there was nothing else to eat at that time. So people were using grass pea as a staple crop. And this is not happening in nowadays. It will not happen. Everybody knows that if they eat any legume as a staple crop for many months in a row, they will get sick. It's completely disequilibrium diet, right? But when we include this legume into an equilibrium diet, I think this problem will not appear. And so we are creating a stigma that is not really there. It exists in other regions of the world, but even in that regions, it's not a big issue right now. So it is a problem when there are big droughts and people don't have anything else to eat. So I believe, and I really like the work that Icarda is still doing a lot on ODAP content resistance, but there are other issues that are being left out that are really important for our conditions right now, and probably don't have anything to do with ODAP, and they also deserve our attention to make it a more attractable crop to farmers. Okay. And I don't have that problem in Portugal. Nobody talks about ODAP in Portugal, the farmers. None. Because there is no records.
Okay, thank you.
So don't create a stigma where they does not exist. I think it's
too complex.
The message.
beta-N-oxalylaphtha beta-diaminopropionic acid. So that's too long. So we have to forget that.
Sure. In Italy too, as there are certain regions where it is traditionally used, and there is very little talk about Lathyrus.
Because it was consumed always in an equilibrium diet. So that is why. That is the most important message we need to pass to our consumers. Eat properly. So eat an equilibrium diet. Don't eat only one stuff at a time.
Sure. Thank you.
Okay, I think we are going to the end of our presentation. I thank again Carlota for her contribution. You're very welcome. Always a pleasure to hear from you. From this month, just a small communication for the group. From this month, we are switching a social outcome. So the Legume Society is not going to be on Twitter anymore. Former Twitter, now X. We are moving to Blue Sky because it's more appropriate in this period. And I see you I see you all there. But as usual, all the information is on our website. You will find us also on Blue Sky. Thanks again, Carlota, and thanks to everybody that was here today with us. I'll see you all next year with a new presentation from Valerio Hoyos Villegas. And where we are, again, we will talk about our related topic, that is what is actually stopping, what is keeping consumer out from a diet that contains more beans and pulses. Okay, thanks again. I'm going to stop
recording. Thank you, Nelson. It was a pleasure.
Sure. Bye.
Bye-bye.